As a scientist, there are really two main areas where I focus most of my energy and attention. The first is spending time in the lab coming up with ideas and doing experiments to test them. A second, perhaps equally important task is to take what we've learned in the lab and try to communicate it to many different audiences, including other scientists or funding agencies or even the general public. And this isn't always very easy to do, as you might imagine. Recently, a friend introduced me to these RSA Animate videos, and I immediately thought that whiteboard animation would be a great style for explaining scientific concepts as well. As my friend Jason and I quickly discovered, it is not as easy to make these things as those cognitive media guys make it look. So in any case, this is our first attempt at it, and what I'm going to try to do is explain a little background about my research, which is in the area of bio-nanotechnology. So your first question might be, what is bio-nanotechnology? We can start by breaking it down. Everybody knows that bio stands for biology, which is the study of plants and insects and animals and basically all of life on this planet. Next up is nano, which is short for nanometer, which is equal to 1/1000th of a meter. Now, I'm always totally amazed by the diversity of life at the macro scale, but something that I think makes life even more amazing and more elegant in a way is that when you zoom into the nanometer scale, it turns out that we're all made of the exact same tiny bits of stuff. Things like DNA and proteins and sugars and fats. Basically, nature has taken just a handful of really versatile building blocks and just figured out how to arrange them in so many different ways to solve pretty much every problem that you can imagine. Now, you can talk about 1/1000th of a meter, and of course that sounds like a really small distance, but it's not necessarily easy to understand how small that really is. So I like to use a US penny as a visual aid to connect the concept of a nanometer to something tangible that you can actually hold in your hands. If you look at the back of a shiny new penny, it probably has some kind of Captain America shield on it. But if you look at a slightly older penny, it shows the Lincoln Memorial. And right in the middle of the memorial, there's a tiny Abraham Lincoln. And if you can imagine tiny Abraham Lincoln's eyelash, it would have about a diameter of 100 nanometers. So next time you come across a penny, take a look at the back, and maybe you could try to imagine that small scale. So now we know bio, we know nano. What about technology? I like to think of technology as what we get when we take all of our knowledge from science and engineering and harness it for some purpose. So in the field of bio-nanotechnology, we're taking the building blocks of life, and specifically the nanoscale building blocks, and trying to invent new ways of putting them together for useful applications. For example, one application of bio-nanotechnology might be to build tools to help us understand how life works at the molecular level. If you wanted to understand how some device works at the macro scale, like an alarm clock, the first thing you might do is get some tools to take it apart and look inside to see what's going on. This isn't really a problem for large objects because it's easy to make the properly scaled tools. But if you imagine shrinking that alarm clock down to the nanoscale and then trying to study it, that's going to be much more of a challenge because most of our tools are much bigger than the things that we typically study at that scale. If we want to understand how nanoscale assemblies work, I think it makes sense to build tools that are roughly the same size. That way it'll be easier to take apart these systems and understand why they work and the reasons why they might break. And looking forward, we might even be able to use these tools or similar ones to fix problems or maybe even make improvements to existing systems. The hope is that we can use bio-nanotechnology to build new tools that will eventually lead to advances not only in basic science, but also in areas like manufacturing and electronics and medicine. So if that's the long-term vision, how do we actually tackle this challenge in the lab? What we do is we take those exact same components that we find in nature and we reprogram them in different ways. And for many researchers, myself included, our molecule of choice is DNA. The reason for using DNA is that many properties that make it useful as an information storage molecule in nature also make it attractive as a nanoscale building material. And of course, everyone in our field owes a great debt to Ned Seaman, who pioneered this area of research, starting with a seminal paper that he published back in 1982. The nice thing about DNA is that it's simple and relatively easy to work with. There are only 2 standard base pairs. DNA has roughly a 2 nanometer diameter, and each base is about 1/3 of a nanometer wide. And the double helix has a pretty simple and regular helical geometry. Furthermore, nature has evolved an entire enzymatic toolkit for making modifications to DNA, such as splicing it together with enzymes called ligases, or cutting it apart with restriction enzymes, or making copies of it with polymerases. It's also really easy to synthesize DNA from scratch. You simply input the sequences into a computer that controls a DNA synthesizer, and in a matter of hours you can have pretty much any sequence you want. And there are companies all over the world that will sell you this synthesized DNA for pennies per base. For the last couple decades, researchers have been using short pieces of DNA to build various shapes. In 2006, Paul Rodeman at Caltech invented a really powerful method to build larger and more complicated shapes using hundreds of short strands that bind to a long template strand. This method is called DNA origami, and it's been the foundation of my own research for the last several years. So what we do is write computer programs to design sequences of DNA such that when we mix them together, they self-assemble into desired shapes with dimensions of 10 to 100 nanometers. Recently, we worked in William Shee's lab to extend the DNA origami method to create 3-dimensional shapes. Looking forward, we're currently using DNA origami along with functional modifications such as protein or nanoparticle attachments in order to start building tools that are precisely the right size and shape for carrying out experiments on the nanoscale. Okay, so that's about it for this basic introduction to bio-nanotechnology, at least as it relates to my own research. Hopefully you guys found this interesting, and if you found it so interesting that you want to know more or contact us, please check out our links, and thanks for watching.